Yeah, sure.
Thank you. Ladies and gentlemen, good morning. Um, uh, Good morning to all the distinguished guests, including Professor DJ Bhatt and Professor Sridhar, who is the editor-in-chief of Myco India Journal of Indian Fungi, fellow mycologist, and all those joining us across the globe. We welcome to Myco Talk 1. This is proudly presented by the Association of Fungal Biologists and Myco India Journal of Indian Fungi. I am Bele Shonai. I am the managing editor of Myco India Journal of Indian Fungi. It will be my pleasure to guide you through today's exciting event. Before we embark on this journey of discovery, it's my privilege to introduce a key figure in the field of fungal biology. Please join me in welcoming Dr. Shashi Rekha, the Secretary of the Association of Fungal Biology. She
Yeah,
will, she will share insights into the AFB's mission and the significance of today's gathering. Dr. Shashi Rekha, the stage is yours.
yeah, yeah. Thank you. Good morning, and thank you, Dr. Shenoy. Good morning to everyone. Good morning, Dr. Priyani, and good morning to all those who have joined in this webinar. And it's a very interesting thing to have collaborated with Myco Asia and Myco India. It's going on very well. I'm actually getting a very good feedback from many places. So, and it's a very interesting thing. To to tell about the Association of Fungal Biologists, we have our president, Dr. Deshmukh here, and we all have started this with a motto of spreading the knowledge of awareness of mycology or fungi among students and general public, so that we are more aware of the processes and the functions of fungi. And the association deals with various kinds of activities like doing nature trails, workshops, conferences, and especially now from this few months, we have in association with Myco India, we have started this a series of lectures, which is very beneficial to each one, and we are able to interact with a number of scientists. And we hope to have more such interactions, especially with the scientists, maybe offline also, if we could arrange for such kind of activities here in Mumbai or anywhere. We already had one conference in December. We definitely want to have more activities where there is a lot of interaction between the students and the scientists in person. With this, I would like to invite Dr. Priyani to go ahead with the talk, as interesting as bioactives from fungi, which is very interesting, and the students will definitely benefit out of it. Yeah, thank
Yeah,
you.
thank you, Dr.
Thank
Shashi Rekha. Madam Professor
you.
Priyani, just give me one more minute. Let me take this
Okay.
opportunity to introduce you to today's audience, because
Okay.
some of us in the audience may not be aware of your work. So for the benefit of such people, I am going to give a small introduction of you to today's audience. Ladies and gentlemen, it's my privilege, to introduce our distinguished speaker for today's Myco Talk. We are graced by the presence of Professor Priyani A. Paranagama, Chair and Senior Professor in the Department of Chemistry at the University of Kelaniya. She is a luminary in the field of chemical sciences. Professor Paranagama's illustrious career has been marked by her tenure as the former director of the Institute of Indigenous Medicine at the University of Colombo, Sri Lanka, where her leadership and research have left an indelible mark on the study and application of traditional medicine. Currently, she serves with the distinction as the treasurer for the year 2023-2024 and as the dynamic chairperson of the Women Chemists Committee. Her past role as the president in the year 2019-20 of the Institute of Chemistry, Ceylon, underscores her enduring commitment to the advancement of chemistry in Sri Lanka and beyond. Ladies and gentlemen, please join me in extending a warm welcome to Professor Priyani Paranagama as we anticipate the wealth of knowledge she will share with us today on bioactive compounds from endolichenic fungi. Over to you, Professor Priyani.
Okay. Thank you very much. Am I audible now?
Yes, ma'am, you are audible.
Okay. Okay. Let me first thank the Microasia Journal, and specifically Professor Sunil Kumar for giving me this opportunity to make this presentation, to share my experience, what I have gathered through the, uh, through doing research on endolichenic fungi. So I selected the, title as The Journey Through the Fascinating World of Endolichenic Fungi, Bioactivity and Secondary Metabolites. So as you all know very quickly that the fungi, play a major role because it's time for us to search for new bioactive compound, because there are many emergence of new diseases and also development of drug resistant to pathogenic bacteria, and also some microorganisms produce new pathogenic the compound and microorganisms, etc. So as we all know that the fungi are important resource of novel natural bioactive compound. The first milestone is, if we all know that it's the discovery of penicillin, which make a that was the first innovation that the fungi made compound. So when come to fungi, like there are mainly three types: endolichenic fungi, rhizosphere fungi, endophytic fungi. However, you all know that in work on endophytic fungi and also rhizosphere fungi, many publications are available. Many there are many reports published on bioactive compound isolated from these two fungi. But and also the importance of that is the thoroughly mentioned after the discovery of this endophytic fungi, uh, Pteromyces and Riana, that could produce taxol that was isolated from the U bark. So therefore, people used to, the scientists and especially the microbiologists wanted to go for new type of, the fungi identification. So the last 2 decades, you must have heard that isolation of endolichenic fungi. This was isolated from lichens. The lichens are symbiotic association of fungi and algae, but these two are symbiotic. But if they are separated, that means they are helping each other. But if they are separated, they can't survive alone. But what is endolichenic fungi means? So like the endophytic fungi, so it was found this, the lichen thallus contain. There are fungi lived in within the lichen thallus, not symbiotic. They are very similar to endophytic fungi. So these fungi named as endolichenic fungi, and what my the work mainly based on these the bioactive compound that are produced by endolichenic fungi. So let me, I'm a chemist, but I work with fungi as well. But because my, this work started when I was in USA, when I was doing my postdoctoral work with at the Center for Natural Product Research and Commercialization with Professor Leslie, and also I got the opportunity to work with the biologist Professor Elizabeth Arnold. So she's the one who isolated this, the endolichenic fungi and trained me as a biologist, right? So then, what we did actually, we collected lichens, right? And they were surface sterilized, and you can see that this is the one that surfaced after surface sterilization. You cut into small pieces and allowed to grow them in mold extract agar, MAE, and MEA plates, and you can see that the emerging fungi. You can see that the fungi are emerging, and these fungi then we slowly, carefully isolated to new plates, and they are named as endolichenic fungi. So, this is how we got the pure cultures of fungi. And then the next attempt was, we didn't know that there were no report in 2006. I started this work, and there were no report on production of metabolites from endolichenic fungi. Therefore, the new research was started, and to see whether these fungi, endolichenic fungi, whether they can produce secondary metabolites. So the first work was started with this fungi Conispora species, which is the host lichen was the carven beard lichen. You may heard of this. Is a very common lichen, Usnea carnosa. And what I did that, the endolichenic fungi from this, the Usnea species was first isolated. And one of these endolichenic fungi is Conispora species that was grown in PDA and PDB separately. So you can see that these are the two plates. And then they were, extracted into ethyl acetate, and then the crude extract was this tested for different bioactivity. So there in that lab, it's well established. It's a state-of-the-art lab that I found in the Arizona University. So what I got the opportunity to see whether it is the active against cancer. So what we did actually, we did the wound healing assay, and we found the ethyl acetate extract forming the very high activity when compared to the the positive control that we use. That's a known the anti-cancer compound that was used, and we found that the ethyl acetate extract of this one can has very high the cancer compound. Therefore, uh, the bioactive compounds that are present in the ethyl acetate extract were isolated, and I was able to isolate many compounds. Some of them are known. The major constituent was herbarine, which is a known compound, but I was able to isolate unknown compounds, uh, uh, and that were isolated first time. That one is the dihydroherbarine, which shows prominence activity against wound healing assay. So this is how it was found, and then and also we were able to identify many known compound. Among them, this was a known compound. That was the first publication as the first report of endolichenic fungi, metabolites isolated from endolichenic fungi, which was published in Journal of Natural Product in 2007. That was an eye-opener for many. And so after that, many research groups started working on, isolating endolichenic fungi as well as isolating secondary metabolites that are produced from, endolichenic fungi. So actually, I completed my postdoctoral work. Then I came back to Sri Lanka. So with I really wanted to continue study, because that Sri Lanka is it's like a pearl in the world, but it's a biodiversity hotspot. We have a lot of the biodiversity, and also if you look at the lichens available in Sri Lanka, so I work with the lichenologist Dr. Gautami Veerakon. She helped me to uh, do the identification of lichens, and it was like she has identified more than 4,000 lichen species, but we wanted to work with few first, because this was the first study on isolation of endolichenic fungi for the lichens present in Sri Lanka, and also we wanted to say the bioactive compound produced. So with that, we did the molecular identification of lichens present first, and then identify the lichens in that. So this is the protocol that we use for the molecular identification of the lichens. And so we use very common lichens, and we use Usnea species, and also the Pseudocyperaria species, that's another lichen, and Palmotrema. And also so we use the same protocol for the same surface sterilization of the lichens, and then we wanted to isolate endolichenic fungi. This is just an example, right? This is how the endolichenic fungi were isolated from different lichens. You see that? So the many fungi are produced in each lichen. This is example for Palmotrema species, the endolichenic fungi isolated. This is example for Pseudocyperia lichen. These are the fungi that were isolated from that. So my the objective was to see whether these the endolichenic fungi, whether they can they have the ability to utilize the substrate. Therefore, what we wanted to first try to grow these the fungi that were isolated in different substrate, and see whether they have ability to utilization of the substrate. So this is example when starch was used, and we found that they have the ability. This is one example I'm showing you, right? They have the starch utilization ability and also pectin utilization ability. These are specific, the protocol that we use to see whether, these fungi can use pectin as the substrate. Lignin, and we use, different tests for lignin because they make different enzymes. One is lactase, and another one is tyrosinase enzyme, and also peroxidase enzyme. So we found that, the different, this is just a summary of the substrate utilization pattern of, the endolichenic fungi that were isolated from these three lichens. We found that, the some endolichenic fungi has ability to utilize starch, cellulose, and also pectin. Some of them have the ability to lactate. So we look at the enzyme activity, and also we finalize, and we got these results, for the substrate utilization pattern of this endolichenic fungi. And then what we wanted is to get the extract of these secondary metabolites. So as what I explained earlier, we used to grow them in the large cultures. Usually large petri dishes were used, like 50 plates were grown at a time, and then they were extracted into ethyl acetate, and then we operate. So usually we do the the kept on a shaker for overnight, and then get the extraction. And that extraction, we first see what activity is present. So these are the preliminary activities that we have done. We check for insecticidal activity, antioxidant activity, antifungal, antibacterial, and also anti-inflammatory activity, and anti-cancer activity. These are the preliminary assays that we conducted for crude extract of this fungal extract. And with that, what we do, so we found that some of them shows very high antibacterial activity, and different pathogenic bacteria, and also we found that some of them shows amylase. We did the the enzymatic inhibition assay to see whether it can be that those extract contain anti-diabetic compound, anti-lipase compound by doing the lipase assay, and also for the antifungal activity, what we use is the Coltotrichum musae, that was isolated from banana that caused the anthracnose disease, and we checked whether this metabolite has the ability to inhibit the growth of this Coltotrichum musae, and also the test insect used for the insecticidal assay is the Calcebrichus maculatus, which this is a stored grain pest, especially in cowpea. It's called cowpea weevil, and damage the cowpea seeds. We wanted to see whether any insecticidal compound are present in these endolichenic extracts. So these are the different insecticidal assays that we use. We use residual fuel bioassay and treated seed bioassay, and also anti-inflammatory assay was conducted using red blood cell membrane ability test, because we assume in this assay, it was assumed that the cell membrane, whether this what we look at is the damage of the cell membrane of the red blood cells, and the red color appearance that is due to hemoglobin that was measured at the 560 nanometer to seek the anti-inflammatory activity. And also anti-cancer activity, we use different cancer cell line, and we wanted to see, whether we use MTT assay or Alama Blue assay to see the anti-cancer activity. These are some of the results. We have isolated many fungi, endolichenic fungi, and extracted were collected, and we have done preliminary bioassays, and this is just to show you the antifungal activity of ethyl acetate extract that were isolated from endolichenic fungi. You see that some of them, they are they show very high activity against this assay, and also similarly we did the insecticidal activity, antibacterial activity was evaluated, and antioxidant activity we used the DPPH assay, and sometimes we did ABTS, and also to look at the producing power we did the FRAP assay. These are common assays. So those assays were done for the purpose of screening, and we have selected most the endolichenic fungi with very high activity. One of them is the Curvularia trifoliate. So that was produced as this endolichenic fungi. This was selected, and it is known to produce this, the curvularin. This is a common fungi, that used to produce this curvularin as the major constituent, right? What we wanted is to, isolate the compounds that are produced as endolichenic fungi. That was the purpose of study. So this is how we did that. So this is the fungi, endolichenic fungi, Curvularia trifoli, that was isolated and also grown in large plates and also extracted into ethyl acetate, and we got the crude extract. So, and this crude extract, we did the partition first in order to, separate the less polar component in hexane extract, and also the moderately polar ones are coming to the chloroform extract, and the most polar component retained in the 60% aqueous methanol layer. So those three fractions, we did the bioassay guided fractionation here. We found the most active fraction was the chloroform fraction. Therefore, we continued our further isolation with the chloroform fraction. So we found that this is the preliminary results that we found for these three fractions. Yes, you can see the chloroform fraction shows the highest activity when compared to the positive control. And also we check the the antibacterial activity. You see that we use the cell the well diffusion assay to get the see the antibacterial activity, we found that it shows very high antibacterial activity. Therefore, we looked at this, we used this Alma Blue assay also. Initially, we did the cell diffusion assay, but since we found it is active, then continue Alma Blue assay against this bacteria to get the promising results. So these are the results that were the found with the antibacterial assay. And also we did the antifungal activity and antioxidant activity with the 3 assays, and we found all these assays, the chloroform fraction has the highest activity. Therefore, it was decided to continue the isolation of bioactive compound with the chloroform action. So, we used the column chromatography and to do the, fractionation, and you can see that we were able to isolate pure compound, and we found that some of them are very high, with high bioactivity, and then these compounds were isolated, and and they were identified using proton NMR and 13 carbon NMR, and also we did 2D NMR in order to get the structure of this compound. And you can see that this is one of the compound that was isolated and identified using NMR activity. So this is, even though that Curvularia trifoli as a fungi that is known to produce curvularin, but what we found when it is isolated as a endolichenic fungi, we found it produces a different compound which is not curvularin, but it is a derivative of curvularin. This was identified, and it is a new compound that was isolated with promising bioactivity. This is the anti-proliferative activity that was found in this. It was named as PPCT01, and this is the results obtained for anti-proliferative activity. We found that it is active for many cancer cell lines against, and also antifungal assay. We found that the MIC and MLC values were also identified. So it has very small, the MIC and MLC assays when tested against Coltotrichum musae, which caused the anthracnose disease, and also insecticidal assay against Calceologous macularis. We calculated the LD50 values, and we found it is 0.064 micromole. For the residue film biopsy, whereas it was 0.096 micromole for that is for the treated seed bioassay. An antibacterial bioassay against the 3 common bacteria, Streptococcus aureus, Bacillus subtilis, and E. coli. These are the values, MIC values that were obtained. So they showed very high activity. And similarly, we compared the the anti-inflammatory activity, but we did not see very high anti-inflammatory activity of against when the standard positive control use was aspirin. When compared to the anti-inflammatory activity of aspirin, this compound did not show high activity. So therefore, but it was concluded that the major constituent of this endolichenic fungi that is Curvularia trifoli, that was a major constituent, shows a high anti-cancer activity, antioxidant activity, insecticidal activity, anti-fungal activity. So and also similarly, we did the identification of the second compound. You can see that. So it is also originated from Curvularia. That's what we heard. It has this basic curvularin structure, and we found it's another new compound that was isolated from this fungi with high and radical scavenging activity. but when compared to BHT, it was not. It has moderate anti, the radical scavenging activity when compared to that one. And these are the results of the overall results of these two compounds that were isolated from the villi. And the second endolichenic fungi that was we work on is the Penicillium citrinum. It's a common fungi that is that but we were able to isolate it from Usnea as a endolichenic fungi. That's the lichen that we use. And so we found that that was also the same work we have done for that this Penicillium citrinum as well. So we took the ethyl acetate extract and then partition, and then we did the bioassay guided fractionation, and we found the chloroform fraction has the highest activity, but we were able to isolate 3 new compounds other than the known compounds. I'm not highlighting the known compound percent, but 3 new compounds were isolated, and they were named as PP, PC 01, 02, and 03. So that out of that, what we found is PC01 has very high antioxidant activity, which is comparable to the positive control, that is BHT, that we use in the assay. So at the same time, since this is a new the exploration of bioactive compound, so we wanted to know whether we wanted to look at the biosynthesis pathway. Therefore, we very carefully we looked at the structures of this compound, and we postulated the biosynthetic pathways of this compound. Some of the compound we could do that, we could propose it. We found that they produce through the polyketide pathway. For an example, this PPPC02 compound, it's a heptaketide, that's a polyketide that initiate the biosynthesis of this compound, and this is the pathway that we proposed and ended up with the final product that we isolated. So, and also the second compound isolated was the previous one. This is the PC02, and we found when postulating the biosynthetic pathway, the second one lead to the formation of PPPC01. So these are very interesting because they have very similar biosynthetic pathway. So these were, and third compound also we found it is coming from again another polyketide, that's a heptaketide that lead to the production of this new compound as well. So, and this is another endolichenic fungi, that is Deltinia escorchi, that was isolated from the lichen. And also we wanted to study the bioactive compound present. So what we found is very strong antioxidant activity. That was the best the antioxidant compound that were isolated so far. So these two derivatives, these are two the derivatives that were isolated from this Aldenia escorchi with very high antioxidant. You can see that antioxidant activity of the pure compound, which match with parallel to the antioxidant activity of positive control. And since we found that these lichens are collected from tropical forest in Sri Lanka, but then we wanted to extend this project to the lichens present in other part of the country, especially Sri Lanka. We have mangrove plants. So we know that mangrove plants grow in very harsh condition, especially they can bear the salty condition. So we wanted to look at the lichens present in mangrove plants, whether they produce other bioactive, other secondary metabolite, may be interesting. So what we thought is, we identified two locations. One is the one area of the area is from Puttlam district in Sri Lanka, because these mangrove plants are available. This is how they look like. And also Nigama area in Sri Lanka. So what we did actually similarly, we collected lichens. These are the lichens that were collected. We didn't see a lot of lichens actually, not like in tropical forest, but limited numbers. But we collected them. Some of them are new lichens, and they were identified again using molecular identification. And then we did the same way. These endolichenic fungi were isolated, and also then the some of so we wanted to see the the phylogenetic analysis of this endolichenic fungi, and this is the map that was prepared. And also similarly, we did the bioactive studies, what is given here. So these are the some we tested we prepared the extract of all these fungi, and then what we did actually prepare the ethyl acetate extract and test the primary activity. So wherever we see that prominent activity, when the isolation of bioactive compound. This is just different bioassay results that were obtained for this one. One of the endolichenic fungi that was identified with promising activity was Neurospora ugdavi. So this Neurospora species that was identified to do the further studies, and we found it has very high antioxidant activity, and therefore we wanted to isolate the compound that are present there. There were many compounds, some of them are known, but I wanted to tell you that we isolated two unknown compound. One was named as neurosporolol 1, and also this is a dimer, and this was named as neurosporolol 2. These two new compounds were isolated with very high, the radical scavenging activity against ABTS assay. You can see that the results of the ABTS assay, and with IC50 value very similar to the IC60 value of the positive control what we use. And when look at the anti-inflammatory activity, we found that neurosporolol 2 has very high anti-inflammatory activity, whereas the compound 1 shows lower anti-inflammatory activity. So these results were again published in the reputed journal. And then the next one that that is again endolichenic fungi that was isolated from mangrove plant, mangrove lichen, is the Purunula bahina. So this is, and also we isolated this compound. It was a known compound, but we found known compound, but antibacterial activity of this compound was not reported before. So we reported for the first time, antibacterial activity of this compound against Staphylococcus aureus. And another compound that was new compound that was isolated is the that was isolated from endolichenic fungi, Sileria side. So this is the lichen that was used to isolate this endolichenic fungus, and which shows the high anti-cancer activity. It's a cytotoxic actually. So this compound that was isolated, which was again identified as a new compound. Then the another area that was the identified is the to get the lichens is from Nigambo area. These are coastal area where you find these mangrove plants, and these are some of the lichens that were the collected, And there we use a different method because we find difficult to isolate all these compounds one by one. Even though we carried out bioassay guided fractionation, we collaborated this project with actually, the NIPER, this is National Institute of Pharmaceutical Education Research in Hyderabad. That was a grant that was awarded to me from the Science and Technology Division of Ministry of Sri Lanka as a Indo-Sri Lanka collaboration project, where we use the LCMS. And what we use is we have 60 extracts that were isolated from 60 endolichenic fungi, and also we selected priority areas depending on the we use the natural product dictionary that is available with the LCMS, and to select the priority areas. So they are we divided into 3 groups, named as priority 1, 2, 3. So if no activity was found, that they were present in what we did, actually all these were subjected to anti-cancer assay, and the extract that show prominence activity was the priority 1, and then the 2, and also those show no activity, we did not touch. Actually, we left them and we concentrated on the lichens that are present in priority 1 and 2. Because that method we found it is easy. So one of the endolichenic fungi that used for the isolation of compound was the Telleromyces pinopilus. We found we collected 3 fractions using the preparative HPLC, and these are the HPLC fractions, and you can see that here 80% purity of this one, but the second fraction what we found is contain many compound. The third fraction contain the 3 major compound. So with that we were we were able to continue the study with the preparative TLC. We found that the 7th fraction 4.5 milligram with the 1 pure compound, and so that compound, with the use of this the dictionary of natural products, we were able to identify. So it's not a new compound, but it was a new known compound, but it shows very high anti-cancer activity. And also another we found that anti-cancer activity is very high when compared to the positive control use. And another compound, similarly, we work on another fraction, and we isolated a complicated molecule, the name as the fraction 7/30. Again, that was isolated before, but first time it was isolated from endolichenic fungi. So this was isolated using dictionary of natural product. We don't want to spend a lot of time because the dictionary help us to see whether it is a new compound. Then we don't want to go for spectroscopic studies with the help of the mass spectroscopy of this compound, and also the matching the mass spectra of unknown compound with the dictionary, we were able to identify the compound. So this was also identified as talromine B, which was isolated from Telleromyces. So these are the predicted compound, and with that prediction we were able to we did the anti-cancer assay and then finalized confirm the structure, but it was a known compound. And likewise that we purified several compounds using this technique, and we were able to isolate many isolate many compounds using preparative HPLC, and also the structures were identified using a dictionary of natural product. That was very convenient, because and also save a lot of time, because they had this high resolution mass spectrometer. Therefore, we were able to match them and confirm the structures. This is how we did that. We got the first substructures, and then we combine them, and come up with the final structure. These are 3 compounds that were isolated from the endolichenic fungi that was isolated from mangrove lichens. So, and the last part of my presentation, I would like to concentrate. We we see that the promising bioactive compounds are produced in endolichenic fungi. So we know that polythene is the worldwide problem. Therefore, we wanted to look at polythene degradation capability of these fungi. So there, we wanted to design the experiment. We wanted to grow them in liquid medium, solid medium, and in the presence of, we use actually low density polyethylene, and they were and the fungi is grown in what we did actually, we selected LDPE, low density polyethylene, and they were cut into stripes and surface sterilized, and then allowed to grow with the fungi. So we selected 2 medium, liquid medium and solid medium. These are the method we use to see the activity of these fungi. We looked at the weight loss, and also we looked at the tensile strength, and also we looked at the ATR, FTIR, and also we looked at the SEM analysis. So, these are we have, as you know, that we had this fungi, database, and these fungi were, stored in the laboratory of University of Kelaniya. So we selected, few of these fungi which show bioactivity, and tested for, degradation, whether they can deteriorate the LDPE in liquid medium as well as the solid medium. So these are the fungi that show the promising results in the liquid medium, and also these fungi are the one they show deterioration of LDPE in solid medium. So so what we did actually, we calculate the weight loss. This is the procedure protocol we use, and these are the results of these are the weight loss percentage that was calculated when they were LDPE treated in liquid medium. These are the fungi species used, and this is the percentage weight loss. What we found is like so these fungi can cause the 10 to 14% weight loss during the period of time. And this is when they were grown in solid, the water absorption percentage, when they were grown in liquid media again. So we found that they have the water absorption ability also. And also we looked at the FTIR spectrum of each of these LDPE strips, and calculate the carbonyl index, CI was calculated. And we found that they show the different CI and higher CI. This is just one example of FTIR obtained for the blue line shows the before the inoculated with LDPE of the FTIR spectra of LDPE before inoculum. Red is the one after inoculum. What we found is after inoculum, after the incubation, after incubation, we found that many new functional group appeared. So through the FTIR results. And also scanning electron microscope analysis, SCM, were done. And you can see that this is the control SCM image of the control strips, LDPE, and you can see the damaged one that we see that after incubation with the, endolichenic fungi. So we obtain good results. And also we wanted to look at the strength of the LDPE. There we use tensile strength. Analysis was done, and these are the results what we found. We see that percentage reduction of tensile strain was calculated, and we found that sometimes the reduce above 50%. So these are the results obtained for tensile strain reduction percentage. And not only that, we wanted to do the the GCMS analysis of this the LDPE cultures that were done, because we did the GCMS analysis to identify and whether they produce any volatile compounds. You know, the GCMS can detect volatile compound, especially whether any alkanes, alkenes, and whether any volatile oxygenated compounds like aldehyde, ketones, whether they are produced. Therefore, they were extracted after incubation with the endolichenic fungi. They were extracted and extracted into chloroform, and then we did the GCMS, and we found that some of them can produce, new, the carbonyl compound and alkanes as well. And also not only that, so we did the other experiment because it was interesting. This was because this endolichenic fungi has the ability to deteriorate, the LD, low density polyethylene. Therefore, we wanted to see whether when they are incubated with this endolichenic fungi, whether any enzyme activity get changed or not. Therefore, we look at this enzyme activity, qualitative analysis and quantitative analysis. Both were done. The enzyme, what we looked at is the enzyme that they use, to deteriorate the substrates, as I said in my first slide. So we looked at lactase, lignin peroxidase, amylase, lipase, so on, the activity of the enzyme. And we found that the enzyme activities were changed. And also we wanted to do the field assay for this one. Therefore, we wanted to optimize the condition for LDPE biodeterioration using spore suspension. Therefore, what we did is we decided to, the minimum, what we wanted to look at what is the minimum volume of should be used. Those studies were done. This is one. This is to decide the minimum volume of spore suspension that should be applied to, get the percentage waste loss, and degree of water absorption was measured with using 2.4 gram of LDPE, and also which contain these are the different suspension that we use, and we studied it. And similarly, we studied what is the best pH for the spore suspension for different, the endolichenic fungi. So therefore, in the study with the biodeterioration of endolichenic fungi, biodeterioration of LDPE using endolichenic fungi, our major finding was, this fungi, Motagunale scabiose, shows the interesting results, and we found that shows the very high, the weight loss, and also percentage reduction of the tensile strength. And also we found the major changes in the FTIR spectra, that is in liquid, both medium as well as solid medium. And also we find the higher carbonyl index. And also you can see that, SCM images shows the damage of the LDPE when grow in liquid medium as well as solid medium. And similarly, we look at the qualitative enzymatic assay using this, the Petri dish colorimetric method. Lactase and peroxidases are the two enzymes that are highly activated when they are grown in LDPE. So we found that these results, and in conclusion, we found this fungi has very high activity to do the degradation and LDPE degradation, and we wanted to develop it as a the product. So we wanted to do more research on this using field assay. So my students are now concentrating on that. These are some of the the pilot scale application that we are going to use for suspension on LDPE strips. If what we wanted to see that what will happen when introduced into the natural environment. So these are things that we, what we have done so far. So, we were able to show that the endolichenic fungi is a promising source of producing novel bioactive compounds. And also we found that, the lichens that are present in Sri Lanka, is a promising source of endolichenic fungi, and also they can produce novel compound. So we did the bioactivity, using bioassay guided fractionation, and we found several new compounds present and isolated, and bioactivity were reported. And also, since it is a time consuming process, we thought that LCMS dereplication study would be the best method to expedite the process and identify new compound. We turned to that. That was done in collaboration with Sniper in Hyderabad, Sniper Institute in Hyderabad in India. And and also we did some in silico studies as well, since time is too long, so I didn't concentrate that. But that was also very interesting. So we will like predict more bioactivity with the new compound that were isolated by using in silico study. So finally, I like to thank funding sources because I got several research grants, especially from the Ministry of Science and Technology, to collaborate with the NIPER in Hyderabad to do interesting study, and also National Research Council, and also National Science Foundation, and also University of Kelaniya. These are my foreign collaborators. This project is successful because I collaborated with many lichenologists, and also the mycologists, because I'm a chemist and I need the help of others. So therefore, I collaborate with them, and I really thankful them for the strong collaboration. And also many people from Sri Lanka, I did the collaboration, and also these are my research students who did the postgraduate work. Some of them graduated as PhD students or MPhil students for that. And finally, I like to thank my family, my husband, and also my son and daughter, and also this little girl is my granddaughter. So they all helped me to go through this journey. Thank you very much. That's all. If you have any question, I'm very happy to answer.
Thank you, Professor Priyani, for that enlightening talk. I hope I am audible to you.
Yes, I am. Yeah.
Yeah. So
I can.
can you? Yeah. Please, could you please stop presenting so that we can have better visual of us?
Okay.
Yeah. Thank you. Yeah. Professor Priyani, now we will move on to the question and answer sessions. I mean, we have a very enthusiastic audience here, and who are keen to interact with you. Can we just spend next 10 to 15 minutes, answering some of the questions, if you are okay?
Yeah, fine. Yes.
Yeah, thank you. Ladies and gentlemen, if you have questions, please raise your hand. I will let you ask. And first, Professor Butt will ask. Professor Butt. Professor Butt, if you can hear us.
Yeah. Um, Thanks, Damodar. It's a very nice talk, Professor Priyani, you gave us, and I'm pretty delighted to hear your amazing talk it is. I have a small question which is not from the secondary metabolites, but I'm curious to know these endolichenic fungi, right? Yes.
Yeah, yeah.
How do they live here? That these ascomycetes fungi, do they preferentially live in lichens or just a casual settlers?
No, they freely
I'm
live
curious to
inside.
know because
It's very similar to endophytic fungi that you see in plant tissues,
okay,
right? So
that's there,
similarly,
yes.
the lichen also they use as a thallus, and these fungi stay inside the thalli. Yeah.
That's very fascinating because that means their host spectrum is pretty wide. Because some of those fungi are very common ones. We see them
Yeah,
as saprophytic
yeah,
fungi. Yeah,
correct.
but it's truly amazing.
I
I'm
agree. Yes.
also wondering whether this kind of host space, do they infuse or induce them to produce some interesting secondary metabolism? We still don't know, but certainly endolichenic fungi are one group which you have shown us wonderful um, their secondary metabolites. Yes, thank you very much. I'm
You're
really
welcome.
pleased to hear your talk.
You're welcome.
Amazing talk. Thank you.
Thank you. Thank you.
The next question is from Dr. Mohan Kumar. Yes, Dr. Mohan Kumar, please.
Yes, ma'am, it was a very great, insightful talk.
Thank you.
My question is on now, almost this endolecanic fungi always live with an algae,
Yeah.
but when we wanted to cultivate that in the laboratory, so I think you have used PDB or PDA, all those types of media. But generally, when you want to grow in the laboratory, won't it be are providing an easy source for the growth, and we are asking fungus to produce a secondary metabolite? Because most of the time, when you see the association, they live in a harsh condition, and also this association maybe it is not present with a less water, present with less water or less other types of metabolite, and whatever the metabolite obtained from the algae, which will help in the growth of the fungus. But where we are growing in the laboratory, we are giving an easy source and we are asking the
Uh,
fungus to produce.
yeah. Can
uh,
I clarify this?
yes, ma'am.
Actually, the lichen is a symbiotic association of algae and fungi. It's a separate, body. But this endolichenic fungi, they have nothing to do with this symbiotic fungi. That is separate, totally separated from that. These fungi, they freely live inside the thalli, right? So we know that these symbiotic ones, if you separate it from algae, they don't produce second metabolites. We have done that study. If it is the lichen symbiotic ones, but these are different. They are just freely live inside thalli, very similar to the endophytic fungi which you see in plant cells, plant tissues, right? So nothing to do with that. So if you isolate, they can grow independently. This is what we did actually.
And also it's fascinating, it produces a lot of pigments. So have you not
Yes.
come across any pigment production
No, they
from the fungus?
produce most of this pigment. We did actually, but we found that most of these quinones, most of them are quinone. They are non-compound. That's why I didn't highlight, right? So they are non-compounds, but unknown compounds only that I showed to you today. They produce different type of pigments. That's true.
All right, ma'am. Thank you, ma'am.
Did I answer your questions?
Yeah. Because I have not
Thank you.
seen that particular entire pigment production in the presentation.
No, I didn't
So
mention it. Sorry,
yeah.
sorry. Yeah,
So
yeah,
yeah,
yeah. That's true. That's
yeah,
true.
yeah.
So the next question is from Dr. Madhavi Tiwari. Dr. Tiwari, please.
Hello. Good morning, sir. Am I audible to you?
Yes, yes.
Yes,
Yeah. First
yes.
of all, ma'am, I'm very thankful to you for such a wonderful research regarding the findings of endolichenic fungi you gave today. Ma'am, I have some very basic questions. First of all, I would like to start my questions from isolation. A very basic question is there, ma'am.
Yeah, yeah.
Ma'am, is there any specific technique we follow during the isolation of endolichen fungi during surface sterilization? It's a very basic question, but we start the other fundamental things from here only. So I want to know that is there any specific chemical we make use during the
No,
isolation of endolichenic fungi?
uh, actually, it's just
Ma'am, Madam Professor Priyani, I think we have lost connection. I will wait for her. Yeah. And audience, please mute yourself. Thank you. I request the audience, please mute yourself. Thank you.
Damodar, I think connection or connectivity lost. I think.
Yeah, we will wait for her. Yeah.
Yeah, we'll wait for it.
It got disconnected.
I'm sorry. I think I got disconnected.
Yeah,
I'm
thank
sorry.
you. Nice to see you back, ma'am.
Yeah. Sorry about that. Right.
No problem. No, it happens. Yeah, no
Yeah.
problem.
So to answer the question that Dr. Tiwari question, so actually the same protocol we use that we use for isolation of endophytic fungi. That's same. That's for the surface sterilization. There's no specific any other treatment we did. It's okay, Dr. Tiwari?
Yes, ma'am. Two more questions I'm
No,
having.
no, no, no. No, not right now. Maybe there are some 10 more people waiting for the turn. Sorry about that. If you have
Okay.
time, we will let you ask at the end of this end
Okay.
of this queue. Yeah, Kirtan, please.
Okay, sure.
Good morning, sir. It was a night session today.
Thank you.
And thanks for providing info about this endolite fungi, ma'am.
Thank
And
you.
my question is about polyethylene degradation, ma'am.
Yeah.
Like coming to, is there any regulatory or safety considerations that need to be like implemented to use this endolectonic fungi for large scale polyethylene degradation, ma'am?
Actually, we did the laboratory scale one now. We are designing the large scale one because we wanted to see that whatever the results that we observe during the laboratory studies, whether you can see actually with the natural environment. That is what we wanted to show. What is your question? Can you repeat it?
Like,
Yeah.
1 second. Like, is there any like coming to superbug for oil spill, ma'am? That will be degraded itself when it can. I mean, when there is a oil spillage in the ocean or like that, ma'am. Like coming to this endolichenic fungi, when it is used for polyethylene degradation, are there any special properties that can be regulate this endolichenic fungi, ma'am?
No, actually, what we did is a preliminary work, I have to tell you. We did not do extensive analysis of this one. And so these are the preliminary work that looking at the weight loss, water absorption, whether FTIR gives different changes, and SCM, whether you find any damages of the LDP, so on. Right, that's all we did. But now one of my PhD students continuing it and in this direction. I can't exactly tell you what the thing should be done for that one, because we are now still doing that.
Okay, ma'am. Fine. It's fine. Thank you. It was an
Okay, you're
insightful
welcome.
session, ma'am. It was a new topic
Thank
about
you.
endolichenic fungi for me.
Okay.
Vaibhavi, please.
Yeah. Hello, ma'am. Good afternoon.
Hi.
It was a very nice session to hear, ma'am.
Mm.
Actually, I had a question which was initially answered by you, that are the endolichenic fungi same as the endophytic
Yes.
fungi? So yeah,
Yes.
they're the same because even I'm working on mangrove endophytes of Maharashtra from
Uh-huh.
India. So
Okay.
I have got the Telleromyces genus. So
Uh-huh.
I mean, that is why I was asking if they are the similar fungi.
Yeah.
And second question was, ma'am, how do we isolate secondary metabolites from the enterolychenic fungi? I mean, is there any specific way?
What we did actually, we collected the fungi. We go in large scale. So, as I said, so like we use 50 plates. Uh, you can use liquid medium or the solid medium as you wish. You can use the PDB or PDA to get the large scale. It depends on the which one gives the large yield, and collected to ethyl, extracted to ethyl acetate, and that's the one, that ethyl acetate extract, because we are interested only in small molecules, because if you go for large molecule, that take lot of time to do the isolation and difficult, but we concentrated isolation of small molecule. That's why they were extracted into ethyl acetate. Okay.
Yeah, yeah. Thank you so much, ma'am. Thank you so
You're
much.
welcome. You're welcome.
Simran Dani, please ask your question. Simran?
Uh, yes, sir. Good morning, ma'am. So
Good.
happy to be able to ask you this question.
Okay.
So I found the LDP degradation very interesting.
Okay.
My question is that in natural habitats, it is unlikely that they might have come in contact with these kind of molecules. So how do you think that they are able to degrade these in vitro?
I think they are, we have done another study we isolated fungi that live in decaying wood,
Mm.
and we did similar work, and we found this similar fungi are present in this decaying wood as well. That's why we wanted to try this. We have a big database. We collected a lot of endolichenic fungi, and they are stored. So we tried, and it worked. It looks like they can, get adapted, and they
Okay.
change, and they use LDP as a substrate as well.
Okay. Thank you so much, ma'am.
You're welcome.
Anil Behra, please ask your question. Anil, if you are around.
Yes, sir. Hello, ma'am. Good afternoon.
Good afternoon.
Ma'am, thank you very much for the insightful talk. My
Good.
question is it required to run a standard compound as a reference when we are analyzing our bioactive compound in GCMS or LCMS, like to match their fragmentation spectra?
Actually, the GCMS, there's a big database that is available. So you can do the comparison. Only thing, if it is a new compound, of course, you have to get the NMR mass spectra and do the spectral analysis and get the structures. So, we did not know, we didn't use standard compound at all because when the structure elucidation is difficult to do that, especially natural products. These are, very rare compound and especially if it is new compound, no standard as well. So all what we did actually, we gathered the all the NMR spectra, and also we did a high resolution mass spectrometer and got the molecular weight and then figure out the structure. That's what we did actually.
Okay, understood, ma'am. Thank you, ma'am.
You're welcome.
Madam Revati Ravi Shankar, please.
Ma'am, good afternoon, ma'am.
Good afternoon.
This is Revati. I'm from Chennai. Thank you so much for sharing your insightful talk on the endolichenic fungi. It was really fascinating and was a wonderful session. And my question is, it's my request, I would like you to throw a light on the pigment production from this endolichenic fungi. Have you worked on this? Have you isolated any pigments from endolichenic fungi? Could you just...
Actually, we isolated several pigments, but we were not interested. Our objective is to isolate novel compounds. So that's why, so we didn't go that direction. So whenever we knew that it is a non-compound, we didn't, it's already reported, then we didn't check. We wanted to go for the isolation of novel compound. So that's why. So we didn't concentrate in that direction. Pigment production, we didn't concentrate. Okay.
Okay, ma'am.
Yeah.
Okay, ma'am.
Okay.
Rishabh Pimenta, please.
Am I audible?
Yes, you
are. Yes.
Amazing session, ma'am. Good morning. Actually, my doubt was you spoke about mutualism being there between fungi and endolichenic fungi. So my question was: is parasitism also like? Is there an aspect of parasitism and maybe take advantage of that to extract secondary metabolites? Like, for example, you spoke about curvularia. I know for a fact that that causes a lot of brain abscesses and a lot of medical issues in humans. So, is it possible to extract the phytotoxins which are generated or the toxins generated and create toxoids to make vaccines? Like, is there a possibility for that? That was my question.
Yeah, that is possible because these fungi, some of them, I know that all these fungi are very common fungi that present in other sources as well. So if you go through the list of fungi I have shown you, right, they are in isolated as endophytic fungi as well. So, but what we found throughout our study, so the secondary metabolites that are produced are not the same what you find as when isolated as the endophytic fungi. Good example, Curvularia trifoli, which was isolated previously and shown that curvularin is the major constituent. But in our study, we found a different novel compound, but it has the same basic structure, but not same as that curvularin. It was a derivative of that, which shows different activity. So therefore, that many people are now working on this area. If you look at the endolichenic fungi, many review papers also coming out because it's an interesting area and new source of bioactive components. Okay.
Yes, ma'am. Thank you. Have a good day.
The last question from Dr. Milan Samarkund from Sri Lanka, who is probably in Thailand now. Milan, can you hear me? Okay, if probably can't hear me, Dr. Manjusha, please.
Sir, I have a question.
Wait, Som. Dr. Manjusha, are you around?
Yeah, I am, sir. Thank you, ma'am, for that wonderful presentation. My question is, have you always isolated, are these associations specific? That is, a specific lichen from which you have isolated only a specific fungi? And is it always one fungi that you isolate from a lichen? Are there more than one fungi
No,
from a...
no, in my presentation I showed you, right? So some lichens produce more than 30 endolichenic fungi. But I saw in literature, some of them, they have presented hundreds of fungi isolated from lichens in literature, if you look through. But I couldn't do that much. But I got, not one or two, many coming out as endolichens. They are really living there, just same as endophytic fungi.
Okay, thank you so much, ma'am. Thank
You're
you.
welcome.
Ladies and gentlemen, the last question from Somdatta.
Thank you, ma'am, for sharing your knowledge with us. Ma'am, I would like to ask a question that about the mycorrhizal fungi. That there are many mycorrhizal fungi which form alliance with plants and help plants to overcome various stresses, both biotic and abiotic. Ma'am, does mycorrhizal fungi also produce the phytochemicals which can be harnessed in for various purposes regarding antimicrobial compound production or and other economically important products?
Of course they can. They also can produce secondary metabolites. Yeah.
But ma'am, there are no. But ma'am, currently the avascular mycorrhizae are not a separate entity. They from association with plants, and there are no methods to culture them separately, like in case for bacteria and other fungal organisms. So can some approaches be made, like bioinformatic approaches, so that we can know about the genes in mycorrhizal fungi which are responsible for the production of these compounds?
We can actually, that's another study that you have to do. We have done this phylogenetic study, and we looked at for one group of endolichenic fungi only. So likewise, in your case also, I think we have to first grow them and see whether they produce secondary metabolites, and then only and see the bioactivity, what directions to do that.
Thank you, ma'am. Thank you.
Thank you, ladies and gentlemen. That was a wonderful discussion we had. I'm sorry I am not able to accommodate more questions now. If you have any more questions, please email to Professor Priyani. I have already shared her email address in the chat box.
Yes.
Any final comments from Dr. Sunil Kumar Deshmukh, President of the Association of Fungal Biologists?
Actually, when we were working with endolichenic fungi, when I was the part of Piramal, that time I have seen some of our papers. Then that time Abhijit Kate was there, who is in Naipar, Ahmedabad. So that's why I was knowing her that she is doing very good work on that. That's why I requested her to make a presentation over here to enlighten the people that, okay, this is one of the field where large of antibiotics are produced, microbial metabolites are produced. And I'm thankful to her that she agreed to make a presentation with very short notice. Thank you, madam.
You are welcome.
I think it's time for me to propose the vote of thanks. Thank you very much, Professor Priyani Paranagama, for spending your Sunday with us. Not only that, you have shared very insightful information and lots of knowledge with the audience today. And today we are richer because you have shared lots of knowledge with us, and most of us will agree with that, and many would probably contact you for further guidance. I
Yeah.
hope you will help some of these young people who are part of
Yes.
our Micro Asia community, Micro India community, AFB community, CBEC community. Thank you very much, ma'am. Thank you so much.
You're
I have one
Yeah.
more thing. If she can make a publication for Micro Asia or Micro India.
I'll try to give a review paper actually, but condensing everything together. So it's nice piece of work actually. If you think it's okay, I can.
Yeah.
Yeah. Thank you, ma'am. And we will let you go now. Thank you very much. And
Okay. Hi.
yeah, thank
Hi.
you.
Good.
And
Oh,
namaste. Yeah.
Namaste.
Yeah. I'm also very thankful to the Association of Fungal Biologists, especially Dr. Suhil Kumar Deshmukh, Dr. Shashireka, Dr. Mahavir Gosavi, for stitching this collaboration. I mean, we have been making lots of good vibes on social media because of all these activities, and people are getting coming towards us, and they're attending our lectures. I hope the lectures are very useful to you, and this is the first talk under AFB Micro India, Micro Talk series. There will be the next talk will be on the 29th. Then next will be on the 7th April. So it will continue. Thank you very much, Dr. Deshmukh, Dr. Shashireka, for supporting us.
Yeah, thank you. Thank you so much.
And I am very much thankful to all the wonderful audience today. Madam, can you please mute yourself, Dr. Shashireka? Okay, thank you so much. Yeah, and I would like to thank our audience. They are wonderful, and they always come up and with lots of joy, lots of curiosity, and they make the session take the session to a different level. And I'm very much thankful to the wonderful people in the audience. With this, I think we can conclude the session, Dr. Deshmukh.
Yeah, sure.
Yeah, thank you, Madam Professor Priyani. Have a good Sunday there in Sri Lanka. And Dr. Deshmukh.
Thank you very much, Dr. Suhil Kumar, and also Dr. Bella, for giving me this opportunity. Thank you.
Thank you. Ladies and gentlemen, namaste. Ladies and gentlemen, I will share the feedback form. Please do share the feedback, submit the feedback. And if you are keen on getting a sub-participation certificate, so that should help you. Thank you so much. Yeah. With this, I just say bye-bye to you. Bye.
Bye-bye. Bye-bye.